So this is the translation data generation. So it's not quite as interesting a title as you'd think, but yeah. So we have the bridging the gap, as we'll try and discuss as we go along. So obviously, with any new development, any novel technology, you have that exciting bit at the beginning, and then during the academic part when you're developing the idea, and then you end up with this valley of death or this gap. To be able to get it through to eventually to a practical solution that can be used in everyday life. So any novel technology faces this challenge of how do you get across this gap, and to cross that gap you need to have enough the data to be able to have confidence, or the confidence in that data is a key component in achieving this goal. So this is kind of where this work package sits. So when we define translational data, the aim of PRISMAP is to say this is data that's applied into the translation research to convert scientific discoveries into practical applications that benefit human health. So this is an aim of PRISMAP, especially in Work Package 11, to develop data that can be used in this translational research to provide confidence in the results that people are using. So this is, so if you're doing your research at the beginning, so you understand, you have confidence in the data that you're generating, that you can look at it, you can then show other people, and then everybody can feel that the numbers you're generating correct, and accurate and robust, which is really important to all of this. So within nuclear medicine, you have the activity dose response relationship. So you have the radiopharmaceutical that you administer to a patient. There's uptake to the tumor cells. So then you have the energy deposited from the radionuclide decaying to the tumor cells. And then you end up hopefully having the death of a tumor cell or some other biological response to it. And this is essentially where we're looking at here. So at the beginning, we have the activity of the radiopharmaceutical, the radionuclide in that radiopharmaceutical, the decay data of that, so they're important components. So that's talking about the Becquerel at this point. And then we have dose, that's the energy, when you're talking about the energy deposit to that tumor cell. And again, decay data comes in there as well. So those are at least straight away 4 factor areas of data that you need for following through this activity dose response. And then finally you have that response. That doesn't have a unit yet. I know there are people trying to define units for this response. So hopefully that will come at some point in the years coming forward. But this is also the biological response, and therefore you want to be able to have some understanding of how that's really important for essentially what you're trying to achieve, which is destroying these tumor cells and activating them. So there's the data, trying to generate data to look at how that process works effectively. So the objectives in the PRISMAP translation data generation work package. So the idea was, provide data that underpins the development of the new radiopharmaceuticals, and to measure and disseminate data across this activity dose response relationship. So with the first work package task, we have the development of traceability for these novel emerging medically relevant radionuclides, and also the decay data that goes along with them. We have determination of, and this was carried out by MPL and CHUV IRA. And then secondly, we have a project work by CERN and Riga Technical Institute to looking at determining the absorbed dose into nanovolumes. And then we have the work by University of Oslo, which was looking at the biological response to quantify and assess this link between the dose and the response, and the relative biological effectiveness. So if we look at activity first, so really everything comes down, like where I come from, MPL, so we're in the National Metrology Institute. So we're all about this traceability, primary standards, and disseminating those standards down to clinics and preclinical researchers along this traceability chain. It's basically a pyramid that goes down, that you work down, and as you go down, the uncertainties get bigger and bigger. So the dose delivered in nuclear medicine, this is a bit different from external beam therapy. So in that, nobody cares about activity because there isn't any. But in nuclear medicine, the dose is delivered is intrinsically linked to the activity of the radionuclide administered. So for the estimation of activity to a tumor, and that's the dose, like you need to know work out the dose. It's based on, you've got two approaches. Most of the time you have the total activity that's been administered to the patient and some understanding of the biodistribution of the radiopharmaceutical, or more recently, you can actually directly measure the activity deposited or in the tumor area or tumor site through quantitative imaging. And in that, you basically have a standard and you calibrate your camera, and then you can determine the activity by imaging from the images you take of the patient. So all of these activity measurements taken at preclinical sites or clinical level typically use a radionuclide. We try not to use dose calibrators because they're not really measuring dose, they're measuring activity. So for the safe use, so in that case, all of the safe use of these and effectiveness of radiopharmaceutical when you're administering to the patient, you need to know what an accurate activity content that you're administering. So one thing to think about with these novel medical radionuclides is these calibrators probably have not been calibrated for these radionuclides because not many people have ever used them in the state we're having them, with this high purity and high activity. And those calibration factors or the results that the calibrators are giving you might not be traceable to any national or international standards. And this can pose a problem where you're putting it into you're measuring it in this commercial calibrator, getting an activity, and then you're assuming that value is correct for what you're using for your research, where it can be very, very different in reality from the activity that is actually there. So this is why it's really beneficial to be able to have these standards and calibrate these radionuclide calibrators so you can have confidence in the activity of the radiopharmaceutical. And you can do this by ensuring those medical devices are calibrated and traceable to national and international standards. But the problem with this is it's not really a problem, but this requires the development of those standards at a National Metrology Institute, an NMI, through primary standards, which can take a long time to develop and are kind of complicated because a primary standard, you can't actually calibrate your detector to the Becquerel in the first place. So you have to do things in a way that you can work out the activity there without calibrating your detector system. So when we're talking about the importance of these standards of activity in nuclear medicine, so when we're talking about this, we're looking at the patient safety and the treatment efficacy, and we can see just very simply how it's used along the whole clinical radiopharmacy pathway. So your activities in the production, supply, radiopharmacy, for determining your activity, the administration, quantitative imaging, and then for your dosimetry. And all kind of concepts, you need to know the activity that is present. And then we come on to decay data. So we're keeping this brief because I don't have that long. So these are the constants of the radioactive decay. So these are really important. These feed into pretty much anywhere you're measuring activity or dose or anything like that. And you can see some examples of where the different nuclear decay or decay data are used in the different processes or different areas of the radiopharmacy pathway. So one of the things one of the projects within PRISMAP that we first looked at was reviewing all of the decay data that was currently available, or the status of the decay data that was available in the nuclear data databases and in literature. We ended up producing 2 deliverable reports, which looked at the decay data for more than 40 radionuclides. From that, we have recommendations for new data studies that are needed for 32 of those radionuclides. Sorry, pardon me. And this is this is basically a good guide for where new studies can be done for these radionuclides and where there are definite needs. So this is kind of a little bit of a summary. So over the last few years, or the process, the period of PRISMAP, MPL and CHUV IRA have been busy standardizing many of these novel radionuclides that have come out through or supplied through PRISMAP, and looking at the decay data at the same time. So between the two labs, we've managed to investigate 14 radionuclides, generating traceability chains for 11 of them, and making submissions to the International Reference System based at BIPM in Paris, measuring half-life measurements, and emission intensities. So one of the things to note about the half-lives and emission intensities, in a lot of cases, these radionuclides haven't been looked at since the 1960s, and through the work that we've done here, that we found a lot of discrepancies, or where there have been significant improvements in the data from the new measurements that have been made, which have been made possible by the purity and the activity that have been made available through the PRISMAP production. So moving on to the dose component. So this was work performed by Riga Technical Institute in Latvia and CERN. So they've been investigating nanovolumes for nanodosimetry. So you can see the benefit that you get from this, where the nanodosimetry would allow for the direct study of absorbed dose at this DNA level, which would be really beneficial for giving you a greater understanding of the interactions of this radiopharmaceuticals at a more granular level. So they've been investigating the use of diamond nanoparticles and silicon nanolayers as potential technological routes towards generating such measurement data. And through their investigations, they've shown that these absorbed doses and nano objects may be measured, but further work is still required to reduce the errors in the measurements of the dose. So there's still a lot of work to do there, but they seem to be progressing that forward, and it could be quite an interesting output. There we go. So then we can move on to the response. I skipped one. Yeah, there we go. So University of Oslo have been leading a lot of this work, or leading all of this work. So understanding the relationship of the absorbed dose to the relative biological effectiveness is essentially a key to understanding how efficient or the efficacy of a radiopharmaceutical. So while there are a number of platforms currently used to investigate this relationship, that the data is limited for understanding how good these current platforms are for the alpha emitters. So a lot of this is a focus. Therefore, they've been focusing on alpha emitters for this work. So they've undertaken a program for alpha particle emitters to develop in vitro methods for estimating the absorbed dose and the RBE, to identify the limitations of the current platforms, and to propose new workflows for more reliable microdosimetry. So the limitations that they've identified, so obviously you get incorrect physical dose estimation, so can be based on a single time point uptake. It assumes a uniform activity and full decay of the radiation site, and just pick a few. So you can also have radon escape, energy loss through membrane. So there's a large number of limitation factors. So yes, and that feeds into how so then you have the problems of relative biological effectiveness and biological endpoints. So obviously biological effectiveness expresses how biologically potent the radiation is, and this is compared against a dose needed to achieve the same biological effect as a reference radiation. So they'll be using X-rays. And there's different ways that the relative biological effectiveness could, or the effectiveness can be derived from. So you have clonogenic assay, DNA double strand breaks, persistent DNA damage, and some other methods. But one of the things to think about is that different endpoints yield different RBE values. So that becomes quite an important consideration to how you can compare how things are working, or making this dose to RBE relationship. So with this dose to RBE, so you have the activity, the uptake, the absorbed dose, biological effect. I've lost my notes. So we have, where we have the RBE for the C4-2P, or they've looked at 2 different cell lines, and it's similar, but you get consistent with alpha particle linear energy transfer. But the RBE is not a fixed value. It varies with the endpoint, the dose, the geometry, and experimental conditions, and therefore we can't consider there's any universal RBE number. And then we have the response from the enhanced. So looking at 3D spheroids to take into consideration crossfire effects. So you get enhanced radiotoxicity in 3D spheroids due to the crossfire effects. So therefore, some different therapies are stronger in 3D spheroids than they are in 2D monolayers, for example. And that is where you have to take account of the crossfire effects from neighboring cells. So in conclusion, so PRISMAP has been delivering new data to help translate new radiopharmaceuticals from the bench to the bedside. The translational data being generated covers this activity, dose, response relationship within nuclear medicine, and Nucleus new standards of activity of radionuclides, and accurate, precise decay data are being determined, which will underpin the dosimetry going forward for these novel radionuclides. And then we have new possibilities for nanodosimetry have been investigated with some success. And there has been a lot of work in investigating this response relationship between the dose-response relationship, and it shows that identical treatments can lead to different outcomes within the same cell line and the radiopharmaceutical. And with that, thank you very much.